Hello friends, welcome back to my channel X and Protein X. In this tutorial video, I'll discuss about the spike and ACE2 interaction. Basically, in this video, I'll discuss mainly the SpikeACE2 interaction in crystal structure form using the Pymol. But before that, just have a quick look that how this kind of spike and AC interaction occurs. So for that, you have to look at this schematic diagram. So here you see this is the schematic diagram of this the interaction. So here is like little bit about this virus. So when this virus is the first the outbreak of this virus, so it was the first, it was replicated in the primary host, so it was the bat, and afterwards it replicated in another secondary host, and thereafter the tertiary host is the human. So human, it was replicated, and that virus was very deadly, and this kind of virus is totally different. Is a lot of mutation in this virus that in the human originated virus, and that is very much different that originated first in the primary host, and this virus is known as the SARS-CoV-2. This virus is higher transmissibility rate, and this is here the if I zoom in this coronavirus, so it looks like this, and here you see this the outside this this lot of spike proteins, and here also this is the membrane. These are the membrane, this brown color part, this outside part is the membrane. Inside it is there is another nucleocapsid, and in this nucleocapsid inside it there is some single-stranded RNA. So you know that SARS-CoV-2 is the RNA virus, so the genetic material is the single-stranded RNA. So now if you come here, this, if you look at this part, so this is the basically the human part. This is the human cell membrane, and human cell membrane here is the ACE2. This is the angiotensin converting enzyme, and it has a lot of function. This is basically, this is the one of the enzyme in the renin angiotensin aldosterone system, and this enzyme involved in the blood pressure regulation. So here in the spike protein that that attach in the cell membrane of the SARS-CoV-2, and this spike protein, they interact with the ACE2. So the spike protein, they utilize this ACE2 receptor to get entry of their genetic material into the human host. So basically after that, when after interaction, they incorporate their genetic material into the host cell, and then has in the host cell inside the human, this there are a lot of replication of this RNA, and then finally it was like a higher rate of this genetic material is inside the human cell. So this is the just a very brief overview of this spike and ACE2 interaction. So in the next slide, I will show the little bit about the ACE2. What is the ACE2? So ACE2 is angiotensin converting enzyme, as I mentioned before, and this is a renin, aldosterone, and angiotensin in this system. So this system, it actually in the kidney, and this is a part of the system that regulates very well and very tightly manner. It regulates the blood pressure. So here what happens here is the angiotensinogen, and it converts to the angiotensin 1. Subsequently it converts to the angiotensin 2, and this angiotensin 2 is the main function of this angiotensin 2 is vasoconstriction, pro-inflammatory, pro-oxidative, and pro-fibrotic. So these are the important function, but main function is vasoconstriction. So angiotensin 2 is the vasoconstrictor. So here the angiotensin 1 is converted to the angiotensin 1 to 9, as well as the angiotensin 2 it converts to angiotensin 1 and 7, and this conversion it catalyzed by S2, angiotensin converting enzyme 2. If you look here, so angiotensin 2 it has the important function is the vasodilation. This is the most important function of this angiotensin 1 to 7, that is converted by the S2 catalyzation. And also it has other some common effect, that anti-inflammatory, antioxidative, antifibrotic, that we looked here, but the main important function is the vasodilation. So this is how it regulates the blood pressure and also the electrolyte homeostasis. So this is the brief overview of this S2. So now I'm gonna move into the main part. So that is the spike and S2 molecular interaction level. So how it interact? So here is the SpikeACE2 interacting amino acid residues, and these residues are important, and these are the main amino acid that interact with the spike and AC2. So basically this amino acid residues are for the AC2 S2 linear structure. So this is the AC2, alright. This is the AC2 amino acid residue structure. But here there are some specific and selective amino acid residue, they interact with the spike protein. So this spike protein, there are some selective, some amino acid residues, they interact with some selective amino acid residues of the S2. And here is showing the red part, it's showing the SARS-CoV-2 virus spike protein that interact in this amino acid residues, but the blue one is for the SARS-CoV-2. So this blue one is at I, E, Q, so this and so on. So these specific amino acid residue are responsible for the spike of SARS-CoV-2 interaction, right? So let's move on to the Pymol. So I will show you in the Pymol that how in the molecular level how this kind of interaction happens. So let's look at the Pymol. So I already opened that the spike and S2 interaction, this crystal structure. I found it from the PDB database. I'll show you that PDB database how. So before that I would like to change it in the manner of the structure. I would like to change. So it's better to change in cartoon format, then it will be very easier to distinguish them, right? And here you see that another things I have to do, that I have to hit the sequence, then I will get this entire sequence of the entire protein. So here is the two different proteins are there, spike and S2, and if you look at this PDB file, right, this is yeah, yes, this is the PDF file that I downloaded. This is that ID. This is the SARS-CoV-2 and S2 complex. So this is the ID, and here you find, if you scroll down further, you can see this is the spike glycoprotein. Spike glycoprotein having the three different chain A, B, and C, right? So it requires in the Pymol when I will make this kind of interaction. So here is the 3 and there is 1, so in total 4 chains are there. All right, so let's move back to the Pymol. So for that, you see here the spike is for 3 chains. So here is one chain, that is the A. So B chains is this is the C chain. So B chain should be somewhere in between. B chains, here is the B chain, you see. So there are A, B and C, 3 different chains. So it's very clear that this part, the left side part, if you look very carefully, so this part there are 3 different chains. You see 1, 2 and 3, the 3 different chains here, and this is must be the spike protein. All right, so only D is the S2. So here is the D. Where is the D? Let me check it. All right, it's the C, number D must be here. Yes, number D. So number D is the S2 protein structure. All right, so now what I do, I would like to change the color of these two different protein structure, then it will be easier to distinguish. So for that I have to select it manually, and it takes like a longer time. So be patient and watch this video till end, then it will be easier for you to get it. So what I do now, I would like to select it. First I would like to select the S2. S2 is not that much big as the spike protein, because spike protein has only three chains in total. So that is a bigger one, but it has only one chain, so that is the small proteins. Right, it's already covered the whole entire area. So you see here is very clear and is very clearly covered this entire area, already selected. So what I do right now, I would like to rename this first, rename it the S2. All right, and also I'd like to, okay, I'll keep it as it is. I will not change the color, so I'll change the color of this one, then it will be better. So now I'll select this one, spike. It is a longer one, so it takes a little longer time. But also there is another alternative way that you can follow as well. Like if you would like to go through the paper that mentioned in that PDF file, so if you go through and if you carefully read that paper, then they mentioned that which chain is actually interact, because not all of the chains are not responsible for the interaction. Only their single chains are mainly they interact with another proteins. So if you go through that and if you check that one, then accordingly you can select that chains only. That will be shorter process. But here I would like to select the entire protein. So it's almost done, already covered. Is oh sorry, it's only one chain I covered only, so it's light is almost done. But for the selection of these two different proteins and and also it is separated by color, it will be better understanding, and also it will for if you present somewhere, or maybe if you would like to if you would like to use this model for your publication purposes, then also it is better to distinguish by color. It's almost done, so I guess is I have here already. So yeah, oh, another chain is here, and this is the last chain. Be patient. It's almost done. This is a more longer time taking. Yeah, it's done. It's almost done. Yeah, it's already done. Alright, so I guess I done already. I think this part I can, but something here is overlapping. What is this? No, it's not. Alright, no problem. Alright, so what I do right now, I would like to rename it. So this is actually okay, so I would like to rename the spike. Alright, so this is the spike protein, and additionally I wanna change the color as well. So I need to change the color red. Alright, red is okay, no problem. Red, no, maybe this color. Yeah, this color is fine. Alright, so this is the two different color, and it's very easy to distinguish two different proteins. So the big proteins is the spike, and the small one is the S2. Alright, so now the job is I have to do another thing. I also here the paper that mentioned here, you see the paper here, the paper and the literature they published, and if you go through this paper, then you can see that they mentioned here the interaction in between SARS-CoV-2 and SARS-CoV-1 as well, SARS-CoV-2 and S2, and SARS-CoV-1 and S2. But here I will focus on only the SARS-CoV-2 because the complex is SARS-CoV-2 and S2. So they already mentioned here SARS-CoV-2 and S2 and this amino acid residue. So I wanna select manually these amino acid residues selectively. So if you wanna do randomly, but I guess is already studied, and if I go through only the selective amino acid residues is better, it will be accurate actually. So I already wrote down that amino acid residues. So I wanna select manually. So from here see the K417, so I can start from the spike. So K417 is here. Let V445 into V445, G446, Y449, Y449, L455, L455 here, F456, Y473, Y473, A475, G476, G485, G485 here, A486, N487, Y489, A490, Q493, G496, Q498, T500, N501, G502, V503, and Y505. So almost is done. So here I wanna do the rename this selection. So what I do, I wanna rename it first. I wanna rename it the spike interaction. Alright, this is better to understand. Spike interaction. Alright, and then additionally I wanna do the manner of the structure that interact in the spike protein, that amino acid residues. I wanna change the manner. So I wanna change it. So from here, as there, as sticks. So I already changed, and also I wanna change the color. Then it will be spectral color. I'm gonna change it to this color. Oh my god. Yes. So it's very clear now. Now what I do, I wanna select the S2. Actually this kind of selection is it is the longer time taking. Otherwise it is not that much long time to take. So I do the S2. The S2 S19, I21, E23, Q24, E25, T27, F28, L29, D30, K31, H34, E35, E36, E37, D38, Y41, Q42, Q42, Q L44, L45, L79, L79, M82, Y83, Y83, 3 to 4. It is almost done, only 3 or 4 left. T3 to 4, T3 to 4, Q3 to 5, G3 to 6, N330, G352, G352 here, K353, G354, D355, R357, A386. So last one, R393, R393 here. Alright, so it's done. So I should rename it first. I can rename it the S2 interaction. S2 interaction. Right. So here I done already. So what I do here, I also would like to change the manner of this structure. it's better to have the sticks and then color. So here already is done, and the color also I'd like to change it. Color I'd like to change to yellow. So it's done. Then you can see, if you zoom it, then you'll be able to see this color. So it's very clear that in the interacting amino acid residues are together. Alright, so what I do right now, I have to do that because you cannot see right now the hydrogen bond that are interacting in between that amino acid residues. So what I do, I have to find the interacting hydrogen bond. So for that, what I have to do, I have to do from the action. So under action, you get this find option, and then under this option you can get the polar contacts. After that, you can get the two other atoms in object. So if you click this one, then you can able to see the interacting hydrogen bonds. So you see very clear that interacting hydrogen bonds are interacting in between these amino acid residues. This is very much clear, you see, and that this all of the amino acid that I selected is all are interacting because these are already studied, right? So if you do it randomly, so it cannot be that much perfect. So you see here this kind of you see here, this is the this structure. Here this is the bond. So here if you can go through the you can and also you can check it carefully, no problem, you can find it. But if you would like to get to know the name of this amino acid residues that are gonna interact with in between, then also you can do as well. What I do, you have to do the under this option you can find one that residues. So if you hit that residues, so then all of the amino acid residues that are interacting, so the name is showing here, is mentioning. Then it will be easier to understand that which amino acid residues are interacting. That's it, guys. So if you would like to use this image, so you can download it and you can save it, and then you can do the further process. So I hope this video will be helpful, and if you like this video, kindly hit the like button, share it, and subscribe my channel. And if you have any comment, so you can write in the comment section. Thanks, guys.